Okay, so we are going to start the session today, and more than happy that we have three big lecturers today, big scientists from different parts of the world. We have one from the Netherlands, Professor Westenbrink, and then we have one from Mexico and America, Professor Gerardo Garcia Reyes, and Professor Julie Chan from Taiwan. So many people in the audience is also widely distributed, around the world. And we have also an example with the speakers. So thank you very much to all of you for being here, and also for taking the time to give this lecture today in session number 10 of the MMD course that is being very successful until now. We have another 9 sessions to run, and then we finish with a beautiful, I'm sure of that, symposia of the students in the last 2 sessions. So let's start this morning session, or this evening or afternoon session, by mentioning to you the program. We have first Professor Garcia Rivas from Mexico, then as a second speaker, Dr. Dan Westenbrink from the Netherlands, and finally at the end, Professor Julie Chan from Taiwan. So let's initiate this session with Professor Gerardo Garcia, who will talk about mitochondria and associated cardiac diseases. Please remember you can ask your questions using the chat or raising your hand. So Gerardo, thank you for being here. Let me just introduce briefly to the people to you. You are a PhD and also a research professor at the Cardiovascular Research Group in Tecnológico de Monterrey. Also, Gerardo is a leader in the cardiovascular medicine research group at the Hospital San Brano in Mexico and Monterrey. He has been working in cardiovascular metabolism for several years. He has a very productive research line in that, and is also involved in the generation of nanostructures and nanovectors for research, and is a co-founder also and a spin-off with a company called Nano4Health. So it's very active in every single sector. He's also a member of the Mexican Research Council and member of the Mexican Science Academy and the National Academy of Medicine. So thank you very much, Gerardo, for being here today. All yours.
Thank you very much, Luis. Yeah, let me share the screen. Okay.
We are seeing to you go up to stop display.
Just try to.
That's fine.
It's fine. Okay, perfect. Thank you, Luis. Thank you very much for the invitation, and say to hello, and thank you to Harry for the space to share some data from our lab in this exciting course of mechanisms of metabolic disorders. Today, we have a session with mitochondria as central organelle for energy production. As you know, we have a significant role in metabolic pathologies. To start, let me say something about the human proteome. The influence amongst all aspects of the cell biology, with particular interest in pathogenesis. Therefore, in identifying and understanding the post-translational modification is critical in our point of view in the study of disease treatment and prevention. As you see in the slide, the human genome proteome is significantly more complex than the human genome. While it's estimated in 25,000 genes, total number of proteins in the human proteome is estimated over 1 million. This estimation showed that single genes encode multiple proteins, first in the genomic recombination, transcription, initiation, and alternative splicing for the transcriptal mechanism that generate different RNA transcript for the single gene. But the increasing complexity in the proteome is related to protein to post-translational modification. This chemical modification played a central role in functional proteomics because they regulate activity, localization, and interaction with other molecules like proteins, lipids, DNA, and cofactors. Moreover, the mammalian proteome is a dynamic and change in response to several factors, such as response to hormones, and posterolateral modification are used to regulate the cellular activity. Posterolateral modifications occurs in the several amino acids that are most mediated by enzymatic activity. It's estimated that 5% of the proteome perform more than 200 types of post-translational modification. These enzymes responsible for translational modification include kinase, phosphatase, transferase, ligase, which add or remove functional groups in the proteins. In additional point of view, in the protein post-translational modification can also reversible depending on the nature of the modification. For example, kinase or phosphorylase donate or remove some phosphate groups. In summary, the analysis of proteins in their post-translational modification is relevant for the study of cardiovascular disease and, of course, another disease. In this regard, the characterization of post-translational modification provides a central insight to the cellular function and underlying physiological, pathological process. With this idea, the agenda for today is talk about 3 recent papers from a group which reveals the diversity, the first for translational modification in mitochondrial proteins using a systematic review, and later 2 experimental papers exploring acetylation as a post-translational modification of mitochondrial proteins in the context of heart failure and pulmonary arterial hypertension, which affects arteries in the lung and consequently the right side of the heart. To start with this part of systematic review, let me introduce that in the cardiomyocyte, which is the muscular cells in the heart, 95% of the energy used for contraction relaxation cycles come from the ATP synthesis by the oxidative phosphorylation in the mitochondria. In summary, this system relies the proton chemical gradient across the inner membrane, which establish a mitochondrial membrane potential known as the proton motif force. This energy is used by the ATP synthase to phosphorylate ADP to ATP. By this reason, inner mitochondrial membrane is a proton selecting and able to restore the gradient through respiratory complex, in particular the electron transport chains, the mitochondrial membrane potential to sustain the ATP synthesis. In this sense, permeability of the inner membrane can change through into a less selective state during disease and stress, in which some of the most relevant components of the oxidative phosphorylation, such the ATP synthase, the adenosine nucleotide translocase, may be recluded in a non-selective pore known as a mitochondrial permeability transition pore. This pore openings allow the movement of molecules across the inner membrane, eliminating the proton pumping, the mitochondrial gradient, and ATP synthesis. Most experimental studies in heart physiology have concluded that the metabolic inhibition, high intracellular calcium, mitochondrial calcium overload, and severe oxidative stress are the main factors driving the opening of permeability transition pore. But recent data suggests that some post-translational modification could also mediate the opening of permeability transition pore through the modification of key components and increase the opening probability, leading mitochondrial dysfunction and cell death. In this part, we conduct a systematic review that involved the first, focusing on the claim the potential constituents of at least the regulators, the proteins regulators of the mitochondrial permeability transition pore, based on information provided for 4 decades of reviewed articles. And second point, we developing a query to retrieve through PubMed all research articles indexed in Medline can report post-translational modification in components, in particular for the permeability transition pores, and cross-referencing to functionally relevant data retrieved for a multiple database of protein post-translational modification. And the third part, we try to integrate the most updated list of post-translational modification in components along with their potential and physiological relevance in cardiac disease. As you see in this slide, we identified as component of a permeability transition port several proteins in the inner and outer space in mitochondria, including the cyclophilin D, which is a proline isomerase, voltage-dependent anion channel, the translocase of adenine nucleotides, the ATP synthase, the phosphate carrier, the creatine kinase in the mitochondria, the exokinase in mitochondria, and peripheral receptor of benzodiazepines. As you see in this figure, we identified a phosphorylation as more reported post-translational modification. You can see in the number of articles that identify phosphorylation in the several components. The second one modification more reported is acetylation that was identified in particular in the protein cyclophilin D. Probably more exciting is in the another figure in which we identify for every component of the permeability transition port several post-relational modifications that includes phosphorylation, of course, acetylation, but and others like oxidation. And using the database for translational modification sites, we identify and communicate, compile several sites specific with clear relevance in the cardiac physiology. Of course, the ATP synthase is the protein which more reported post-translational modification in several sites, in particular sites settings for phosphorylations. Later, we try to define and integrate the most post-translational modification in this component in physiology and pathophysiology. In particular, in cardiac disease, we observe that ubiquitination of exokinase, the mitochondrial exokinase, is associated with decrease of cell apoptosis and diabetes. The voltage-dependent anion channel during ischemia reperfusion, nitration is altered, the channel function and decrease the glycosylation was found to exacerbate ischemia and reperfusion. In heart failure, we observed that phosphorylation mediated GSK was associated with opening of permeability transition port and apoptosis. In the translocase, the adenine nucleotide translocate in ischemia reperfusion, nitration was associated to the opening and apoptosis. We identified that succinylation drive decreased expression, driven by decreased expression of SIRT25 is associated with post-ischemic contrail dysfunction and increased the infarct site. In the case of ATP synthase, we documented that was found succinylation that induced by decrease of expression, by the decreased expression of SIRT23 was associated with contractile dysfunction and increase in infarct site. And in heart failure, we identified that hyperacetylation induced the decreased expression of by the decreased expression of SIRT23, and hyperacetylation of several ATP subunits increases the interaction with cyclophilin. Finally, cyclophilin D, which is a central mediator in the permeability transition port opening, has been associated particularly with hyperacetylation related with decrease of activity of SIRT23. In additional, some SNE translations was founded as critical points for MPTP activation. Will we present some data regarding the particular papers, experimental papers in which obesity, metabolic syndrome, and diabetes induced in the cardiac muscle an increase in reactive oxygen species and a mishandling in calcium. This is a consequence with altered mitochondrial activity associated with the permeability transition port opening, particularly regulating cyclophilin D as a protein. This altered mitochondrial activity is associated with metabolic inflexibility, energy depletion, decreasing contractility function, which is clearly associated with heart failure. To introduce, let me introduce this protein, sirtuins, are deacetylase, which remove the acetylase group from several proteins. These sirtuins depends of the metabolic state in the cell. In particular, are dependent of NADH. In this sense, contribute to modulate or communicate signals of our overload of nutrients, particular in obesity. Another interesting data is that sirtuin knockout mouse develop cardiac hypertrophy and fibrosis, and the loss of sirtuin aggravate mitochondrial dysfunction and heart failure. With this, the objective of this first paper is demonstrate that sirtuin-3 downregulation in chronic metabolic changes has a central role in inducing cell dysfunction and energetic impairment in cardiac function. The experimental design is where use human cardiac tissue collect from the humans, and in the time of cardiac transplantation, we obtain samples from lean and obese patients. In addition, we use an animal model for obesity using sucrose and follow the contraction and relaxation in an isolated heart, and we perform experiment in isolated mitochondria. As you see, the difference, the more clear difference in the samples for the human patients with heart failure is the BMI. The BMI for non-obese is 23, and for the obese cohort, there was 34, which indicates that there are obese patients. Using these samples, we purify mitochondria, and we identify in heart failure patients 46 down regulation of sirtuin-3, as you see, and as a consequence of the loss of activity of this deacetylase, we observe an increase in the acetylation profile. In this, with these changes, we perform some correlation study, and we identify, as we expect, a negative correlation between sirtuin and acetylation in the human heart. But more interestingly, we identify a positive correlation between BMI and acetylation. And for the clinical point of view, we identify a negative correlation between sirtuin-3, a BMP, brain-natriuretic peptide, is a biomarker for prognosis in the patients with heart failure. With respect with the animal model of obesity and metabolic syndrome, as you see, the animal showed obesity, increase in adipose tissue, hyperinsulinemia, hypertriglyceridemia, and hypertension. With these animals, we observe in particular when we perform echocardiography and some data from isolated heart, we identify failure in the diastolic failure. We identify a reduction in ejection fraction, but it's very clear associated and very correlate with the human phenotype, showing diastolic dysfunction. We purified mitochondria. We identify the increase in the gene expression of the GCN5L1, which is acetyl transferase. But more interestingly, the sirtuin protein expression was downregulated in the obese group, and the consequence was probably a major acetylation profile. In particular, we observe an increase in the profile of cyclophilin D acetylation that was increased in the obese animals. To demonstrate that these changes in acetylation in cyclophilin have a relevant and physiological role, we explore the calcium retention capacity as a measure of permeability transition pore opening. And you observe in the obese animal, inducing by calcium, the calcium release, opening of permeability transition pore. We observe that the mitochondria for obese animals were prone to opening the permeability transition pore compared to control animals. And we say, we observe using cyclosporine A with a blocker of permeability transition pore, that is the main mechanism responsible for a cardiac dysfunction. In this point, we observe that conditions such as obesity induced downregulation of sirtuin, which modified the acetylone profile in mitochondria, in particular with cyclophilin D, resulting in an increase in permeability transition pore opening and cardiac dysfunction. Patients with obesity exhibit more lysine hyperacetylated proteins associated with EMP and less sirtuin expression. But we think that it's necessary the mitochondrial therapeutics approach to reestablish sirtuin expression or activity, not only for the potential therapy for heart failure patients, but also to prevent cardiovascular disease in several cardiovascular conditions. In this regard, we explore in our group several compounds that modify the activity or expression of sirtuin-3. We use quercetin, we use oroxylin, which have a very nice activator of sirtuin-3. But for today, I will be present data using this interesting molecule, trans-resveratrol, that induce activity and expression of sirtuins in the heart. The activation of regulation of sirtuin by resveratrol, this compound that we found in wine, grapes, dark chocolate, etc., have a potential, has a role increasing the activity of sirtuin, in particular sirtuin-1, but it probably is the same for sirtuin-3, an increase of the activity binding directly to the N-terminus site, increasing the affinity for the protein for the substrate. The mechanism of sirtuin-3 has not been fully described, but potentially some data suggests that is very similar for sirtuin-1. The rationale for this experiment is excitation contraction coupling is need for a proper function in the ventricular myocyte. We use a model of right ventricle failure, which is associated with pulmonary arterial hypertension. As you showed previously, sirtuin overexpression reverse a pulmonary arterial phenotype, and sirtuin knockout mice develop spontaneous pulmonary arterial hypertension. The objective of this study is characterize the functional, structural, and cellular alteration caused by pulmonary arterial hypertension in cardiac and lung tissue to evaluate, describe the mechanism to protective, the cardioprotective properties of resveratrol, a sirtuin activator, in a model of rat model of pulmonary arterial hypertension. The strategy in general is the functional structural changes associated with this model of monocrotaline, histology, electrocardiography, remodeling, and markers. And we purify an isolated cardiac myocyte to explore the calcium signaling, and we purify mitochondria to explore mitochondrial dysfunction. Our model is just a single dose of monocrotaline that in 42 days induce pulmonary arterial hypertension, and we use 20 milligrams per kilogram in the rat for every day to explore the potential cardioprotective effect of resveratrol. We don't identify very clear changes at the lung pulmonary vasculature. We don't identify pulmonary occlusion significant effect and vessel diameter changes with the use of resveratrol. But there was very interesting that we observed that resveratrol prevents cardiac pathological remodeling, reduce fibrosis, reduce hypertrophy. In particular, we observe a significant reduction in the volume and normalize the volume in rats treated with resveratrol. In this condition, we observe that pulmonary arterial hypertension increase the protein acetylation and decrease, of course, markers of hemodynamics in the heart. And we explored the function on cell shortening and calcium handling in isolated fibers of the heart. And we identify in particular that the pulmonary arterial cells from the pulmonary arterial hypertension model show a more decreased shortening, and the uptake of calcium in the sarcoplasmic reticulum that was reduced significantly. But the use of resveratrol normalized the reuptake of calcium, as you say in this quantitative analysis, in this sense, it's important show that the cell relaxation is the most highly consumed mechanism in both the calcium removal by the sarcoplasmic reticulum ATPase and the myosin releasing activity depending of the ATP. In this sense, we explored the mitochondrial function as a producer of ATP, and we observe a reduction in the mitochondrial membrane potential with reduction in oxidative phosphorylation and the respiratory control ratio. And again, we observe that the resveratrol preserves the mitochondrial functioning, in particular, reducing the permeability transition pore. In this sense, we observed that mitochondrial acetylome, there was increase in pulmonary arterial hypertension, and pulmonary arterial and resveratrol reduced and normalized the profile of acetylation in the mitochondria from these animals, possibly related with the increase of expression and, of course, activity of sirtuin-3 and changes in the profile of cyclophilin D acetylation. In this regard, we observed that in pulmonary arterial hypertension, there is an increase in cyclophilin D hyperacetylation with sense to the opening of permeability transition pore, with induced failure in energetics and, of course, calcium mishandling, reducing the cell shortening, but in condition in which resveratrol induced the expression of sirtuin, they decrease the acetylation of cyclophilin D, reduction of ATP, permeability transition pore opening, and increase in ATP, and probably contribute to the correct handling of calcium and cell shortening in the cells. Finally, activation of C2 prevents the energy capacity of the cells to carry out the contraction relaxation cycle by preventing hyperacetylation of components of MTP. Thank you very much for your time. Here are our collaborators from the Metabolic Research Institute, Tec de Monterrey, Institute of Cardiology in Mexico, and of course, all the people that perform experiments in these papers. Thank you.
Thank you, Gerardo. Thank you very much. That was an impressive amount of data presented in the talk. Thank you very much. We have some questions from the people. Here is Professor Ferreira from Uruguay. Thanks for the talks. I wonder if you have explored post-translational modifications in the mitochondrial KATP channel, as it is a cardioprotective channel that is quite related to cell metabolism and the modulation of MPTP.
Yeah, I think this is very interesting. For now, we didn't see this channel, but I know that have a lot of roles, particular with post-conditional and preconditioning in the heart, but we don't have data. We don't check the systematic review was only in the components of permeability transition pore. But thank you, Gonzalo. There was a very nice idea to continue the study of post-translational modification.
Thank you. I know Gonzalo Ferreira is a very good channel study physiology, so you may get in contact later. It's very nice.
Thank you.
And then another question from Marcelo Cornejos. He's in the Netherlands now. And he said the hypertrophic heart is associated with poor regulation of intracellular pH. What is the evidence between the activity of mitochondria and the regulation of intracellular pH? And second, what is first? What is first, the impaired mitochondrial function or the alterations in intracellular pH?
It's a very nice question. The first point, the contribution for mitochondria to maintain the intracellular pH, I think this very regional and is compartmentalized in the matrix, in a membrane space. I don't really see that data that the contribution of mitochondria could be crucial for maintaining the pH, intracellular pH. But I don't really know. Probably if we know that the pH in the inner membrane is very regulated by the several components, in particular the respiratory change that mobilize protons and And I think that I'm not really sure if the mitochondrial impair is associated very clear with alter rate of intracellular pH.
Yeah, but on the other way around, yes. I mean, the intracellular pH may alter the mitochondrial function because the proton transmembrane distribution of protons, will limit the generation of ATP. Yes. Okay. Thank you. And there's another one, Christian Cortez Rojo. Nice talk, Dr. Garcia. Is the HFC-23 activity stimulated by resveratrol related with an increase in NADH levels in mitochondria?
Yeah, we not measure in these papers, but recently we are explored the ratio between NADH, because NAD is activator of CYP23, as you know, and we currently have experiments with a model of heart failure with preserved ejection fraction, and I hope very soon have this data. But at this point, I don't think we don't have this data. But I'm really true that the ratio between NADH is modulated, of course, by the pathology of pulmonary arterial hypertension and heart failure.
Okay. Another question that this will be with a short answer, please, because we have almost only 1 minute. Paola Valero from Chile, thank you very much. Could this treatment with this analog of resveratrol be considered for other diseases that have a strong connection with CFD stress? Like, yeah, I don't know.
Okay. It's probably we exploring with another particular oroxylin A and other molecules that we connected with oxidative stress. But in this data in particular, we don't find changes, really huge changes in oxidative stress. We identify more changes, in particular in post-relational modification.
Mm-hmm. Is it possible to have, I mean, have you read or something about this molecule and cancer, for example, the oxidative stress? Yeah. Are they using it?
No, not yet. But...
Okay. The last question is from Shahid or Sahid Aziz. It says, excellent talk. Do the ROS has a permanent impact on mtDNA? Does exposure to ROS cause changes in mitochondrial DNA, like some mutations, for example?
Yeah, of course, it's very reported that the mitochondrial DNA is very sensible to reactive oxygen species, and probably is in some chronic disease is associated with changes and failure in the mitochondrial DNA. But in particular, we don't check the integrity of the mitochondrial DNA in this condition, under this condition.
Okay, excellent. Thank you very much, Gerardo, and thank you for the people, for the question to the people. We are moving now to the next speaker, is Dr. Dan Westenbrink. He's at the University of Groningen in the Netherlands, and he's a cardiologist dedicated to translational medicine also. He has a PhD and postdoctoral training, is registered as a clinical pharmacologist, and specializes in heart failure and cardiac ameliorate. He has been studying the causes of myocardial energetic deficiency in heart failure. He has received many grants from the Netherlands Heart Foundation, for example, and also from the Dutch Organization of Scientific Research, ACICO, and VENI, which is very well known there. And of course, from the European Society of Cardiology also. It's an associate editor in several journals, and particularly from the European Journal of Heart Failure, for example. So Dan, thank you very much for your time, and the screen is all yours. Thank you.
Well, thank you, Luis, for your kind introduction. I hope that you can now see my screen.
Yes.
So today I would like to talk to you about the potential role of ketone bodies as a fuel that could fix the failing heart. It's a line of research that I've been working on for the last 5 years, I believe. And what I see every day as a cardiologist specialized in heart failure is that despite all our efforts, a lot of heart failure patients still suffer from energy loss. And this is most apparent when they climb the stairs or try to exercise. And it's a strong motivation for me to try to alleviate this suffering. Because we have a lot of drugs for heart failure, a lot of drugs that are intended to prolong life and to keep the patients out of the hospital. But one of the most important complaints by the patients themselves is that they are restrained in their day-to-day activities. So there's still lots and lots of suffering. And I think what already has been covered in this course a bit is that the heart is an organ that requires a lot of fuel, 30 times its own weight every day to pump 10,000 liters of blood. It consumes 6 kilos of ATP and can only store enough ATP to last 3 heartbeats. And it goes without saying that energy production is critical for the heart. And this is exemplified, for instance, by the main cardiac disease, myocardial ischemia or myocardial infarction, where a loss of energy creates myocardial death. And in a normal heart, there is a tight demand-supply equilibrium where most of the energy is produced by mitochondria. In heart failure, there is a major problem with energy production. Regardless of the cause of heart failure, energy production, mostly in mitochondria, is insufficient. Put in other words, and I think you've seen that around quite a lot, the failing heart is considered to be an engine out of fuel. This is not new. Actually, it was first described in 1937 in Galveston, Texas on a conference. Before that, they thought there was too much energy, but when they calculated it again, it was not enough. And this has been a line of research that we haven't solved until now. Because if we look at the current therapies for heart failure, all the medication that we give, like beta blockers, ACE inhibitors, ivermectin, even SGLT2 inhibitors, but also LVADs and CRT devices, are all intended to reduce the energy demands of the heart. But they do very little on the energy supply. And what I believe is that if we can restore cardiac energetics by giving better fuels or more fuel, we may be able to restore the energy equilibrium and maybe even cure heart failure. This is a very simplified schematic of the concepts, the current concepts on what happens to cardiac metabolism. And for the sake of time, I would just summarize this, is that in the beginning, the failing heart is unable to take up and process fatty acids for fuel, which is the main cardiac energy source, even in the failing heart. But when heart failure progresses, also glucose uptake and oxidation is reduced. There is a slight increase in ketone oxidation, but together it's not enough to restore ATP levels, and ATP levels are reduced in the failing heart. This is something that you can just measure in a failing heart with a simple kit. And based on the dogma that glucose is more oxygen efficient than fatty acids, because fatty acids are related to ROS production in the heart, a lot of efforts have been devoted to either reducing fatty acid oxidation in the heart with several types of drugs or stimulating glucose oxidation. And despite decades of research, there is not a single drug targeting cardiac metabolism in heart failure specifically. And what I would like to do, and what I would like to focus on, is trying to see if we could change it a little bit and focus on the other cardiac fuel source, ketone bodies, and if we could increase this to increase cardiac energy production. A very short word about what ketone metabolism actually looks like in the normal situation. Well, ketones are endogenous fuels produced by the body under conditions of fasting or hemodynamic stress, where there is adipose tissue lipolysis, and the free fatty acids from adipose tissue are transferred to the liver, where they are converted into ketone bodies. Acetoacetate or beta-hydroxybutyrate are the metabolically active ones. And because the liver itself cannot produce ketones or cannot consume ketones, they are transferred through the blood to the organs that can process them. And virtually any cell can process ketone bodies, but the main ketone consumers are the brain, heart, and the skeletal muscle. It has been known for a couple of decades that in patients with heart failure, there is an increase in the amount of circulating ketone bodies. And these are the studies that have been done in 1996, where the first discovery was that there is more ketone circulating in the blood. And also in the right panel, you can see that after a glucose clamp and a controlled fast, the closed circles show that ketone body production is really increased under these conditions in heart failure. More recently, and this is unpublished data, we looked into a database of patients that were admitted for acute heart failure and followed up for 30 days. And when you compare the ketone body levels at admission with the levels after recompensation, you can see a significant drop in circulating ketone bodies, indicative of the fact that ketone body production is increased during episodes of acute heart failure. What we also observed recently is that in patients without heart failure, but with an ST-elevated myocardial infarction, during the acute stress of an MI, there's a marked increase in circulating ketone bodies, but also after 24 hours, there's still an increased level of ketone bodies in the circulation compared to 4 months. And what was quite interesting is that patients with the highest ketone body levels in the circulation also had the biggest infarcts and the worst ejection fractions at 4 months, clearly showing that there's a link between cardiac dysfunction and ketone body production. Furthermore, we also observed that there is an association between circulating ketone body levels in the healthy elderly population and a risk for developing heart failure. And quite interestingly, this association between ketone bodies and the risk of developing heart failure in the future was strongest in women. So what I just showed you is that there is an association between ketone bodies and heart failure in patients with patients. But what is the consequence of this ketogenesis? Because of course, if something is up in heart failure, it could be bad for you. And for the sake of time, I won't show you all the details, but there was a quite elegant study performed by Horton from the University of Pennsylvania using mice that are incapable of oxidizing ketones in their heart. And they clearly showed, using multiple models and multiple lines of evidence, that if you are unable to consume ketones in your heart, if your cardiomyocytes cannot eat ketones, you're worse off once you develop heart failure. This means that ketogenesis in heart failure is thought to be an adaptive response. So what we thought that this might be very interesting to use therapeutically. And the next question that we asked ourselves was, what are the opportunities to increase ketogenesis in heart failure? How can we induce this? And at the time, SGLT2 inhibitors were only drugs for diabetes. And what we saw is that patients with SGLT2 inhibitors, diabetic patients treated with SGLT2 inhibitors, had very much increased levels of ketone bodies during fasts. So we thought that might be something that we could use for heart failure as well. So what we did is we used the SGLT2 inhibitor, empagliflozin, in a rat model of heart failure as a proxy to induce ketone levels. And we treated rats either early or late with empagliflozin for a period of 12 weeks. And what we used is a heart failure model of permanent coronary artery ligation to induce heart failure. And as you can see from the pictures below, we were quite successful in inducing large myocardial infarctions, which you can see here in blue. And while there was no difference in the size of the myocardial infarctions between the animals treated with empagliflozin early compared to shams or the ones treated with empagliflozin late, we did see a marked increase in circulating ketone bodies in all animals treated with EMPA, both shams and MIs. This was paralleled by a similar increase in ketone excretion through the urine, so the increase in ketone levels was robust. And what was also robust is that we saw a significant increase in left ventricular ejection fraction in mice or rats treated with empagliflozin, both early and late, and that there was no difference between both treatment arms. And when we looked at the change from baseline in left ventricular ejection fraction, it was quite striking that the normal progressive decline in left ventricular ejection fraction, which we see in this model in the blue line here, was completely halted by empagliflozin treatment. So we were able to preserve cardiac function with this drug. And this was associated with an increase in the expression of the ketone uptake receptor and also both enzymes involved in ketone body processing. And finally, empagliflozin was able to restore cardiac ATP levels, possibly through ketone bodies. Of course, this is indirect evidence that ketones can work because SGLT2 inhibitors have a lot of different functions, and it's still not exactly clear how they work, although they have been registered for heart failure. To look more directly at the effect of ketone treatment in heart failure, we did this exact same experiment, but now we treated the rats with a ketone ester in their chow. And we also started either before the MI surgery or 2 weeks after, making an early and a late group. In the left panel, you can see the effect of ketone esters on myocardial infarct size. And again, similar to SGLT2 inhibitors, we did not see any effect of early treatment on myocardial infarct size. So it's not an effect on the MPT board, for instance. But what we did see, and this was really striking and much more pronounced than what we observed with SGLT2 inhibitors, is that treatment with ketone esters resulted in a marked increase in left ventricular ejection fraction, both in the early and late group. And again, when we plotted the change from baseline in left ventricular ejection fraction, we saw a major increase in left ventricular ejection fraction in the rats treated with ketone bodies, while in the control group, we saw the similar decline in left ventricular ejection fraction, which we were accustomed to. And similar to what we saw with the SGLT2 inhibitors, we also were able to detect a significant increase in myocardial ATP concentrations in the ketone-treated animals. These are data from our group, but obviously there's more people working on this. And what is always reassuring is that people are able to reproduce your data in larger animal models. And what you can see here is the effect of ketone treatment in a porcine model of heart failure with reduced ejection fraction, where ketone treatment through infusion resulted in a marked preservation of left ventricular ejection fraction and an increase in cardiac output. More recently, Deng et al. Also showed that ketone treatment and empagliflozin treatment was able to improve cardiac function, or in a HFPEF model, or preserve cardiac relaxation, I must say, in a HFPEF model, but that this was not due to an energetic effect, but this was due to an effect on acetylation. So ketones are probably working through multiple mechanisms, and in different models, some mechanisms might be more important. But when we look at patient data, there's not a lot yet, but there's a lot of ongoing clinical trials. I'll show you just a few. Here in the left, we can see the effect of an acute treatment with beta-hydroxybutyrate in patients in the cath lab, where a cardiac blood sampling allowed the authors to calculate the percentage of ATP generated through different substances. And these patients were fasting, I must say. So there was not a lot of glucose consumption in the heart, which is here in gray. But what you can see clearly in the red, in the bars, is that giving ketones to these heart failure patients resulted in a marked increase in the percentage of ATP produced from ketones. So if you give ketones to these patients, it is converted into ATP. And similarly, if ketones are given IV in the cath lab, there is a marked increase in cardiac output over time. And dose-dependent increase has been observed by Nielsen et al. Already in 2019, indicating that these data from rats and pigs are probably translatable to humans. And the last bit of human data that I'm going to show is a very recent study where patients were given one ketone ester drink, and before and after an echocardiogram, they did an echocardiogram and then gave a ketone ester drink and measured left ventricular ejection fraction, global longitudinal strain again. And what they could see is in these patients, giving them one drink of ketone esters already increased left ventricular ejection fraction. And I'm talking about patients, but it's actually not a patient study, but a study in healthy participants. And already in healthy participants, there was an increase in cardiac function observed. We are currently testing the effect of ketone esters on ATP production in patients using an elegant MRI technique, where patients are in a scanner exercising on an MR-compatible bike. We can measure ATP production in real time. Hopefully by early 2022, we'll have the results. And another line of research that I'm really interested in is trying to understand how ketones are increased in heart failure, because we might tap into that mechanism to provide an endogenous treatment strategy. But ketones are much more than just a fuel. They have an effect on endothelial function, oxidative stress, HDACs, mitochondrial function, and a lot more. So there's a lot more in store for ketones in heart failure and other diseases as well. So in conclusion, ketone bodies are increased in patients with or at risk for heart failure. Ketogenesis in heart failure is protective and amenable to therapeutic interventions. And I believe that exogenous ketones are an attractive method to deliver ketones to the heart, but future studies should determine whether this will result in meaningful clinical benefits. A word of thanks goes out to a lot of local, national, and international collaborators from all over the world. Thank you very much.
Thank you, Dan. Very nice. Very interesting to see all the patients' data correlating with the experimental assays. Very nice. We don't have any question now at the chat, but they will come. There was one people raising their hand, but not anymore. So if we don't have questions now, they may come during the next presentation, and for the end we can answer the question. And I wonder whether the ketone levels elevated, increasing the ATP in the heart, are due to some specific mechanism, or that you may know in animal models or cell models, or I doubt in the patients. But what do you think about ketones and signaling molecules that we know might be regulating mitochondrial function for the generation of ATP, like endogenous nucleosides, particularly in the heart?
So it has long been known that ketone bodies, in particular beta-hydroxybutyrate, are strong regulators of HDACs, right? So there's a lot of evidence for that, but all the evidence in the heart so far is circumstantial. And what we're currently doing is using these PDH1 knockout mice to try to tease out if it's a fuel or not. And so far, we haven't reached that conclusion yet. But it's something that we can use knockout models and specific studies for that. Obviously, there's also opportunities for in vitro research, but that's not something that we're looking specifically.
And the increase in ATP in the heart, is it because more generation of ATP, or because reduced degradation of this macroergic compound in the surface of the endothelial cells?
Well, I think, you know, the most obvious conclusion would be that there's more ATP production, right? The increase in ATP in this model, where you see a progressive decline in ATP production in normal situations, is much too great to be driven by a lack of degradation. I think that's so far is clear. But what is also very interesting is that although ketones can become the dominant fuel, if you infuse Langendorf hearts with ketones, different compositions of ketones, fatty acids and glucose, if you increase the ketones high enough, there's almost a linear relation between the amount of ketones that you put in and the amount of ketones that are consumed and produced as ATP. But what we did observe in chronic treatment is that by giving ketones and also by giving SGLT2 inhibitors, it also restored some of the machinery for the uptake and processing of fatty acids and glucose as well. So there is some reverse remodeling, and if that is specifically a mechanism that is through ketones, or it's due to the reverse remodeling or the prevention of progressive cardiac decline that we have induced by ketones, I don't know. So it might be circumstantial, or it might be a secondary effect due to the ketones themselves.
Yeah. There are two questions in the chat now. One is a very general one, same from Paola Valero. Are there any side effects with the use of this compound in human studies?
Well, that's a very important question. I can be very short. The most important side effect is that it's also a vasodilator. So if you infuse it, there is some degree of hypotension that can be observed, but only at very high doses. The other thing is that in order to increase circulating ketone levels to a degree that we think would be sufficient to use as a fuel, you have to drink a lot of very bad tasting ketones. Ketones taste very poorly, and there's almost nothing that you can do to improve the taste. And this is something that is shared by researchers in the field as a major drawback throughout. So, but the volumes that you need to give are just too big to put in a pill or other ways of delivering it. So that's the main side effect at this point.
Right. And the last question from Maria Mendoza. Excellent presentation. And is there evidence of reduced ketogenesis in patients prior to heart failure?
No, that's actually really interesting suggestion that there might be a reduction in ketone production while going into heart failure. And no, the evidence that we have so far is that if you are at risk for developing heart failure, your ketones go up. And what I didn't go into for the sake of time is that there's a lot of stress hormones that can increase ketone production, that induce lipolysis and increase ketone production in individuals that are not fasting. So I believe it's the other way around, that if you are stressed and are at risk of developing heart failure, your heart or your circulation starts to promote ketone production by the body.
Okay. And because we have a question from one of the speakers also, Gerardo Garcia, he says, thanks Dan for the talk regarding the use of SGLT2I. Do you think that the beneficial of ketone bodies is the same for, can you read that, HFREF or HFPEF patients?
Yeah, so basically SGLT2 inhibitors are the major breakthrough now in the treatment of heart failure, because it's the only drug that's currently proven to be effective in patients with heart failure with preserved ejection fraction. Well, we have a lot of drugs for heart failure with reduced ejection fractions. We only have SGLT2 inhibitors, and it works for both types of patients, and the pathophysiology is quite different. And what we know from animal studies, and there's only one real detailed study from the group of Rong Chang on the effects of ketones in HFPEF models. And what we see there is that the mechanisms are totally different. So in HFPEF, it's absolutely not working as a fuel. It's working through acetylation of different components of mitochondria. So it's a more direct mitochondrial effect, and it's more remodeling than it is working as a fuel. And also the evidence that there is a reduction in ATP production in the hearts of HFpEF patients is less robust than it is in HFrEF patients. So it's probably a different disease, and it's probably not as effective.
Okay.
Or it may be as effective, but it's working through different mechanisms. That's what I believe.
Yeah, that's right. Thank you very much, Dan, for your talk and for the answer to the questions and comments. We're now moving to the last seminar this morning, and it's going to be given by Professor Julie Chang. Julie is at the Changun Memorial Hospital in Taiwan, and she holds a PhD, and currently is a distinguished chair professor at the Institute for Translational Research in Biomedicine at this hospital. She studied autonomic physiology. She's one of the models in the world for physiology per se, and she has been, she's in fact the immediate past president of the International Union of Physiological Sciences, IUPS, and we just had a very nice meeting, and she was the leader in that. She has been acting as editor in several journals, including Journal of Physiology, American Journal of Physiology also, and her research is oriented in neural regulation of the cardiovascular functions, with focus on brain oxidative stress, nitrosative stress in pathophysiology of the hypertension, and also studying now, of course, developmental programming of adult hypertension. So thank you very much, Julie, for being here, for taking the time, and for staying here with us when we know it's so late in Taiwan at the moment.
Thank you so much, Luis. No problem at all. I'd like to thank you for organizing this course and inviting me to participate in this session. So like you mentioned earlier, that we are in session 10 of this course. It's almost halfway through this series of presentation and courses. So I thought you might have already learned a lot in terms of basic knowledge and current events in this field of cardiometabolic syndrome. So what I would like to do is to share with you our experience of applying those knowledge we have learned from other people and from other study into our research to understand to study the role of mitochondria in fetal programming of hypertension that is associated with metabolic syndrome. I don't think I need to go any further to introduce to the audience, particularly the grad students and young people, of the developmental origin of health and diseases. This concept is actually indicating the risk of a number of chronic chronic non-communicable disease like metabolic syndrome, hypertension, kidney disease, heart failure, and obesity and other cardiometabolic related diseases may have their origin even before birth. And this concept of developmental origin of health and disease was developed from the seminal work by the late David Barker, who demonstrate, like showing this slide in one of his articles published in 1990, that the highest blood pressure that could be observed in adult, whether it's male or female or man or woman, are those born with underweight during their infancy. And this finding has been thereafter been supported by others, both from animal and from human study as well. And according to this concept, we now know that the susceptibility to the development of disease in adult life could be increased by exposure, including not only just nutrition, perinatal hypoxia, gestational diabetes, toxin during pregnancy and lactation, medication, and even environmental toxin or chemicals during fetal life, infancy, or early childhood. So, so this is really the influence of maternal exposure to all these insults or stress that will lead to the disease development in offspring. And in the interest of time, I'll just focus my presentation on overnutrition and share with you how we address the question and design animal model to study the role of mitochondria. And this actually started about 10 years ago. And during that time, we already know that there are several mechanisms that contribute to the developmental programming, to maternal malnutrition. Those include the epigenetic modification of gene encoding protein that are involved in cellular metabolism, energy metabolism, and of course the changes in the nutrient sensing pathway in the peripheral system, as well as in the central nervous system and the insulin resistant. All this have been proposed to be the underlying mechanism to the development of obesity or metabolic syndrome of developmental origin. And one key component that are involved in all these mechanisms is mitochondria. So about 10 years ago, we started our project trying to understand what is the role of mitochondria in developmental programming of metabolic syndrome, particularly in hypertension that Luis mentioned that we have been studying for many, many years. So we generate several models of maternal overnutrition model to study the metabolic syndrome in adult offspring. And one of the model that I'd like to share with you, or I will focus in my talk today, is the maternal high fructose diet model. And the model is generated by feeding the mother during gestation as well as lactation with a high fructose diet. The control animal or control mother receive normal diet. And the offspring, once they immediately after weaning, they will return to normal diet. And we observe the development of cardiovascular function and hemodynamics and other molecular signaling until the age of 12th week. So this is the animal that we use, but before anything, we need to make sure that this animal is working. So what we did is have these two groups of animals, and the offspring from the mother exposed to high fructose are HFD offspring, and those are the NDO offspring. And we measure their metabolic parameters. In this slide, you can see, if you look at the focus on those two bars, you can see the fasting glucose triglyceride is increased in the HFD offsprings. The glucose level is also increased. The insulin level, plasma insulin level is increased. The HOMA index is also increased, indicating insulin resistance. At the same time, the HFD offspring also have a higher blood pressure. So all these major traits of metabolic syndrome that are observed in human DEM can be also duplicated in this animal model. So we then have in our hand a good model to address the question that we are interested in. The blood pressure is regulated by humoral, neural, and hormonal factors. As an autonomic regulation person, that our lab has been interested in looking at the brain control of blood pressure, particularly in the area in the brainstem known as rostroventral lateral medulla, why we're interested in this nucleus or in this area is because this is the area that is actually the premotor neuron that's sending sympathetic outflow from the brain to the vessel, to the heart, and to the kidney to increase the heart rate, to increase the constriction force of a vessel, and to secrete all this angiotensin and other from the adrenal gland and as well as the kidney. So the RVLM, activation of RVLM increase sympathetic outflow and increase blood pressure. And we have published a series of a study and demonstrating that if the tissue is under oxidative stress, particularly in this brain region, will increase sympathetic outflow, which results in blood pressure increase. So first of the question we ask is, we see the blood pressure is increased in the HFD upstream, then whether the RVLM is activated and whether it's under oxidative stress. So what we did was measure the lipid oxidation using the NDA as an index, and can clearly see at the age of 8 weeks or 12 weeks, the HFD upstream clearly have lipid oxidation. In addition to that, we will also measure the ROS, tissue ROS level, and quantify it with a higher level of ROS in the same age time in the HFD upstream. We all know that nitric oxide is another major player in the regulation of blood pressure, but in this animal model, we didn't see any change in NO level, at least not in the RVLM region. So we see the oxidative stress in this brain region may play a determined role or important role in the development of hypertension of fetal origin. There are several ways tissue may generate ROS. In general, there's a membrane bound NADPH oxidase, there's mitochondria in the cardiovascular system, the uncoupling of NOS, the enzyme that is responsible for the production of NO, as well as xanthine oxidase, are all involved in the production of nitric oxide, particularly superoxide in tissue. So we are interested in whether in the HFD upstream, the mitochondria may be play a role in tissue oxidative stress that we observed in this brain region. So what we did was we measure in situ the ROS production in the mitochondria. First, we use we measure the superoxide in the RVLM in situ, and you see those red fluorescents are actually indicating the ROS, tissue ROS level in the RVLM. Then we use the mitosox, which is more specific for ROS in the mitochondria, and use another marker for the neuron. If you merge them together, you can see that those cells are highly labeled with mitosox, indicating in the RVLM of HFD adult, the mitochondria are actually under oxidative stress. The mitochondria electrotransport chain plays a role in redox of homeostasis under oxidative phosphorylation. And during the process, the electron might leak from the inner membrane of mitochondria into the matrix, where they can react with oxygen molecule to form superoxide. So if the mitochondria electrotransport chain enzyme activity is impaired, there's a higher chance that the electron will be leaking out from the inner membrane to the matrix and resulting in superoxide or ROS production. So what the next step we did is to measure the electron transport chain enzyme activity. And we measured the NCCR, which is the enzyme responsible for the electron transport between complexes 1 and 3, and SCCR for complex between the enzymes between complexes 2 and 3, and CCL, the cytochrome C oxidase activity. clearly, I think you can appreciate from this slide that both the NCCR electron transport between complex 1 and 3, as well as the major complex 4, the enzyme activity are significantly impaired in HFD upstream in this brain region. At the same time, if we measure tissue ATP level, you can see there's a significant decrease in tissue ATP level. Again, we measure the mitochondrial ROS level, and we can see the ROS level is increased. So the ROS level increase could be resolved from the impaired in mitochondria electrotransport chain enzyme activity. To confirm this, we use a mobile electron carrier coenzyme Q10, which has been demonstrated by us and by others as well that work through the electron transport chain enzyme capacity to increase the enzyme capacity so that the ATP production could be preserved. At the same time, we can see the ROS production in mitochondria is also significantly alleviated, indicating that this ROS increase in RVLM of HFD upstream may likely to be the consequences to the impairment of mitochondria electrotransport enzyme activity. Of course, once the ROS is generated, the cell has very strong antioxidant defense mechanism to balance the overproduction of superoxide and other reactive oxygen species. In the RVLM of HFD animal, we measured the major antioxidant for degradation of ROS, including catalase, superoxide dismutase, and glutathione peroxidase, particularly the SOD2, which is located in the mitochondria. And we can see in this, under the HFD, in HFD upstream, the SOD2 catalase, their protein expression are significantly suppressed, but we see an increase in glutathione peroxidase protein expression. So it's not universally decreased. We have protein, which the antioxidant, which protein expression is suppressed, but others are increased. To make sure that the antioxidant activity is actually perturbed under this condition, we measure the antioxidant activity, the total antioxidant activity in this nucleus, and found that in adult HFD, indeed, the total antioxidant capacity was significantly suppressed. So together, these data suggest to us that in the RVLM, the brain region that is important in neuroregulation of blood pressure, the oxidative stress, we can observe that under the HFD animal. And this could be the result of both the decrease in antioxidant activity, as well as some of the antioxidant protein expression, as well as increase in mitochondria superoxide production or ROS production, because the reduction in the inner membrane of the electron transport enzyme activity. So there's a decrease of bioenergetics, meaning decrease in ATP production. We also did the same experiment in the heart of HFD animal, as well as kidney. Both are damaged. You can see the ejection fraction is impaired, as well as the kidney function is also damaged in HFD animal. Some of the antioxidant also responsible for that, but others are for the heart as well for the kidney, for the HFD injury in the HFD upstream. In addition to that, we also take an omics approach to identify potential transcriptome that are either upregulated or downregulated because of maternal HFD exposure by the use of whole genome RNA-seq analysis. So this is done in the postnatal day 1, and we took the whole brainstem because it's too small for us to isolate this specific nucleus. But if we look at the whole brainstem of HFD offspring in postnatal day 1, we can identify 29 genes with the transcriptomes among those 29 genes are upregulated and 32 genes are downregulated. Among the top 10 downregulated genes, 6 of them are related to mitochondria, either their function, morphology, and their structure and the synthesis. And one particular gene that really caught our attention was this cytochrome C oxidase subunit 7C. If you remember, in my previous slide, I showed you that cytochrome C activity was suppressed. So we were very interested to see whether this downregulated CCO7C expression are related to the downregulation of its activity. So what we did first was we took the brain tissue out and we did a very simple Western blot to see the expression. And indeed, we can see particularly the CCO subunit 7C expression is downregulated. I mentioned that post-translational modification or epigenetic modification play a very significant role in the developmental programming of metabolic syndrome in adults. Then there are several ways that DNA methylation and histone modification and MIR are all involved in epigenetic regulation of gene that encoding proteins for example, for the mitochondria activity. So we study every arms of the, or every element of this epigenetic modification, but in the interest of time, I'll particularly show you the DNA methylation. What we did is we take the premotor region of mitochondria, CCO7, and did a methylation versus non-methylated DNA and found there's a significant increase in the ratio, indicating there's hypermethylation of this in the premotor region of this mitochondria CCO gene. And we know that the DMMT, the DNA methylation transferase, is mainly responsible for DNA methylation. And in mammals, there are 3 active DMMT family, DMT1, 3A, and 3B. And we can see in HFD animal, the DMT1 and DMT3A expression are significantly upregulated compared to the brain region of the HFD offspring. To further demonstrate the causal relationship of DMT activation is responsible for the hypermethylation. What we did was we used the mini pump, osmotic mini pump, to chronically or for infusion the DMT inhibitor 5-ASA into the 4th ventricle to inhibit DMT. And under this condition, we can see the hypermethylation of mitochondria CCO subunit 7 is dose-dependently reversed. At the same time, we can see the CCO subunit 7 expression is also back to normal or compared to the offspring, indicating DNA hypermethylation of CCO subunit 7C particularly could be a reason that causing CCO activity being suppressed and resulting in ROS production. At the same time, we can see the CCO activity is also partially preserved under the treatment of 5-ASA. So this piece of data indicating to us that the increase in ROS production in the mitochondria of HFD animal could be due to a hypermethylation of CCO7 subunit 7, which suppress the CCO expression and inhibit its activity. And this is partially responsible or engaged in the decrease in bioenergetics and the increase in ROS production. So the next question we would like to ask is why maternal HFD could cause DNA methylation in a brain region of the HFD offspring. We know that the cell has a specific sensing sensor to modify or to to sense the changes in intracellular as well as extracellular nutrient change. And this energy sensing pathway are very important for metabolic cellular metabolism. For example, in the liver, if there's a high fructose diet, this will activate the major nutrient sensing pathway. One is the AMPK, the other one is SIRT1, which is also discussed in the first lecture, that in cardiac heart failure situation, here, once you activate, you inhibit lipogenesis, increase the fatty acid oxidation, and inhibit the gluconeogenesis, and which then will keep have impact on the cellular metabolism. And the skeletal muscle as well, if there's calorie restriction or under starvation, these two major nutrient sensing molecule will be activated, resulting in consequences of changes in cellular metabolism. In the brain, because the brain depends very heavily on cellular metabolism for their neuronal function, so there are lots of area in the brain region. They have this nutrient sensing neurons located in the brain, and in the brainstem, RVLM is actually the one particular area where the nutrient sensing neuron are located. And previously, we have demonstrated together with our collaborator that the major nutrient sensing molecules, for example, AMPK, SIRT1, as well as the PGC1 alpha, participate in developmental programming of metabolic syndrome via their ability to program the mitochondria in terms of their biogenesis, the production of new mitochondria. So we were interested to see, in addition to the impairment of mitochondria bioenergetics, which is decrease in ATP production, would maternal HFD have influence on the mitochondria bioenergenesis in their offspring? So we take a look at the protein expression of those three major nutrient sensing molecules. The phosphorylated AMPK versus AMPK is significantly suppressed, indicating the activity of AMPK is downregulated. At the same time, you can see the SIRT1 expression is downregulated, and the PGC1 alpha protein expression is also downregulated in this brain region of the HFD offspring. Among the three of them, we particularly interested in the PGC1 alpha because it is a transcription factor that regulates the mitochondrial transcription factor TFON expression. And TFON is a very important mitochondria protein that is engaged in the production of mitochondria DNA. So it's involved in the mitochondria biogenesis. And in this brain region of HFD animal, we found the TFON expression is actually suppressed, and the DNA copy number of mitochondria DNA copy number is also decreased. So together, it suggests to us that the HFD offspring in this brain region, the major nutrient sensing signal or molecules are that their expression are downregulated. One of them, the PGC1 alpha, because of the downregulation, will causing a downregulation of a major protein that is involved in mitochondria DNA production, which is TFON, and resulting in a decrease in mitochondria biogenesis. And we already demonstrated that in the RVLM of HFD animal, the mitochondrial bioenergetics is also suppressed. So decrease in mitochondria synthesis, as well as once it's synthesized, its function is also suppressed. Together, this will maintain a sustained increase in ROS in this brain region, as well damage to the mitochondria. And it is reported that when the the activation of AMPK will actually phosphorylate the AMT1, the enzyme that is responsible for DNA methylation. And this phosphorylation is actually has an inhibitory effect on the DNA methylation activity. So if you have increased AMPK activity, we'll have decreased DNA methylation activity. In this case, we see the opposite. We see the AMPK activity is actually decreased. So in this sense, we'll decrease the AMT1 phosphorylation in the serine 730 position, and this will causing a decrease in DMT phosphorylation, and this could be the reason that we see a DNA methylation being increases in the HFD animal. So together, we think this is both the impairment in the biogenesis as well as bioenergetics involving epigenetic regulation and impairment of nutrient sensing signaling are responsible for the augmented accumulation of ROS in the mitochondria of HFD animal. The next question we want to ask is how maternal HFD could cause offspring to have this cellular consequences and the decrease in mitochondria activity. We're still in the process in answering this question, but I'd like to share with you some of the very preliminary data that we see in addition to high insulin level in the offspring, the plasma, as well as the tissue leptin level in the RVLM are actually increased. So we see a hyperleptinemia in the HFD animal, as well as in the brain region. And at the same time, if we look at the leptin receptor, either at the messenger RNA level or at the protein level, there's actually being suppressed, indicating there's a high level of leptin, but a lower level of leptin receptor, indicating there's a dissociation between these two signals, as well indicating a leptin resistance. The consequence to that is once the leptin binds to its receptor, it will activate the AMPK kinase, which will phosphorylate the AMPK. In this case, we see the AMPK kinase activity the protein expression is suppressed. At the same time, is AMPK kinase activity is also suppressed. So in this brain region or HFD offspring, the leptin level is high. There's the leptin resistant, so there's the decreased AMPK kinase activity. This may be responsible for why we see a decrease in AMPK expression and its activity under this condition. So all this identification of the signaling pathway and identifying the cellular processes is very nice, but we think it's very important to identify or to see whether we can reverse all this process, and if the reversal have any protective effect in terms of developmental programming of metabolic syndrome, particularly hypertension that is associated with it. So we see the HFD because hyperlipidemia in the offspring and in causing resistance and inhibit AMPK phosphorylation. And this could be the reason, one of the reasons that we speculated why we detect the DMT phosphorylation is downregulated. So to see whether we can protect or we can alleviate the whole processes with some activator on the protein that we identified, we designed 3 different groups or different experiments. One is to apply resveratrol or metformin to mother during lactation, and the second group is the same treatment to offspring at young age, and another group is at adult. At young age is about after weaning between week 3 and week 6 or week 7, and in the adult is after week 8 until week 12. So this is all daily application of resveratrol or metformin. Again, in the interest of time, I'll show you the following slide of our result on the treatment with resveratrol. First, let's take a look if we give oral intake of resveratrol to HFD mother. Can see in the offspring, this is given to mother, but we look at in the offspring, you can see the hypertension or the increase in blood pressure is protective or reduced. At the same time, the plasma norepinephrine level is also reversed, indicating this is worked through the central nervous system, or at least worked through the sympathetic activity to reduce the plasma norepinephrine level. But if we look at the metabolic parameter, if we give resveratrol to mother, there's no protective effect of the plasma increase in the plasma glucose, plasma insulin, or the plasma triglyceride level. So although it protects the hypertension, but it's no effect or no beneficial effect, at least under this dose, which is 20 milligram per kilogram per day to mother, to the metabolic syndrome in the HFD offspring. But however, if we apply the same dose of HFD per body weight to the offspring, you can see if we apply the young age, which is age between 3 and 6 weeks, can see all these metabolic parameters are corrected by this treatment. You can see there the hypertriglyceridemia is protective, the insulin level is reversed, glucose as well as HOMA index are all there are beneficial effect on those metabolic parameters. But if the same drug is applied to the adult offspring, there's some protection, but not to the insulin level or to the HOMA or to the insulin resistance. So depending on the time when the resveratrol is given to the HFD offspring, whether it's young or adult, they may have differential protection or protective effect or beneficial effect on the cardiometabolic syndrome. Then how about blood pressure? If we apply the resveratrol to the young adult, again, you can see this is increase in blood pressure from week 6 until week 12 in the HFD offspring, because there's gradual increase in systolic blood pressure. This is measured under a conscious condition using the telemetry. And if we give the resveratrol, you can see clearly that there's increase in the increase in blood pressure is significantly being suppressed. The same treatment to adult from during the week 8 to 12, again, the blood pressure is high in the HFD offspring without treatment, and the treatment can partially reverse the increase in blood pressure. But if you look closer, the protective effect of the anti-hypertensive effect of resveratrol given to young HFD animal versus adult HFD animal is much, much stronger or greater. And then we go back to all these molecules that we have identified previously. So once again, resveratrol to mother, can see they have protection in terms of DNA methylation. You can see the hypermethylation of cytochrome C subunit 7C7 was protected, and the activity, CCO activity is preserved. But if we look at the biogenesis, the resveratrol treatment to mother does not seem to have any effect on the impaired mitochondria biogenesis induced or programmed by the maternal HFD. Still, we see the T-fol number is suppressed and the DNA copy number is also lower compared to the ND group. But if we treat the animal the young animal with resveratrol can see the major nutrient sensing pathways or molecules, their expression are reversed. The DNA copy number is preserved, and the DNA biogenesis is also preserved. If we look at the bioenergetics, the CCO activity, which is suppressed in adult animal due to maternal HFD, is the complex, the enzyme responsible for electron transport between complexes 1 and 3 are preserved. The enzyme activity of CCO is preserved. The ATP production is back to normal, and the mitochondria ROS production is also alleviated. So depending on the period of time that the treatment to either mother or young adult or a young offspring or adult, the beneficial effect seems to be different, and this may be related to their differential effect on the mitochondria biogenesis versus mitochondria bioenergetics. So altogether, the current conclusion or the we have at this point is that depending on when and where you apply the treatment or the intervention to mother or to offspring at different age, in this example that I have just presented to you, resveratrol to mother will protect the bioenergetics, but it will reprogram hypertension. That means they will protect the programming of hypertension associated with metabolic syndrome to maternal high fructose diet. But if we apply the same treatment to the offspring at childhood or even young age, they will have beneficial effect on both bioenergetics and biogenesis. This will reprogramming hypertension as well as metabolic syndrome. In the same treatment to adulthood, we also see the protective effect, but I think you can appreciate that the protection or the reprogramming capacity or the influence to hypertension as well as metabolic syndrome are much smaller than if the drug is applied to the child or at a young age. So the early intervention is important than the delayed intervention, which may not have the same protective effect as when the drug was delivered in the young age. So finally, I'd like to thank, particularly in this program, the clinician that I have the privilege working together in our hospital and the young colleagues in our institute. And I thank you very much for organizing this course. I think this is truly, a motto of IUPS, that is physiology without border. And this course is actually a reflection of this spirit of IUPS. With that, I thank you very much for your attention.
Thank you, Julie. Thank you very much. Thank you to you for being here and for supporting this course from the IUPS and for the rest of the world. In fact, we have many students from different countries, and that was the main aim of this course. So thank you very much for that. We have several questions, and I will try to go fast. I have one from Ana Elias Lopez, and the question is, well, excellent and very interesting work. How is metabolic dysfunction transmitted to offspring? I think you showed that. And it's a metabolic dysfunction transmitted through the germinal pathway. Do you know about that?
But this is something that really interesting. We see that the question is whether how mother influenced the offspring. Is that the question?
Yeah, I think it's the transmission of the mother influencing the offspring.
Yes, I think the placenta definitely play a major role, and we have collected the placenta as well as the blood from the cord, and we're trying to see whether there are markers that we can link the maternal influence to those signals that we identify to the placenta as well to the offspring. At the same time, we're also looking at the microbiota that is being affected by high fructose in mother, and see whether there's also have some role in the nutrient sensing pathway, and as well as some metabolite that in the offspring that could be influence the nutrient sensing pathway. We identify this nutrient sensing seems to be a key player between mother and child, but how this being activated, how the maternal high fructose diet may influence this nutrient sensing pathway, maybe the gut microbiota, placenta, and cord blood will give us some hint. We're still working on this part. Okay.
Then Marcelo Cornejo from the Netherlands, he says, does impaired mitochondrial function alters angiogenesis in pregnancy or during pregnancies with consequences for fetal development?
Mm-hmm.
And the second one is it possible that this is a potential cause of increased high blood pressure in young people?
Okay, for the second question first. No, it's not, because this is not consequent. The changing brain is not consequence to the high blood pressure, because we use vasodilator to correct the blood pressure, to lower the blood pressure in the HFD offspring, but still we can see the tissue oxidative stress and all this mitochondrial dysfunction. So it's not the blood pressure, high blood pressure trigger the oxidative stress in this brain region. And the first one is, I didn't catch the first one. Question is,
does impaired mitochondrial function alter angiogenesis in pregnancy with consequences for the fetal development?
Oh, no, this is very likely to be one of the possibility, but we haven't had any chance to study that yet. Yes.
All right. Then there is another one from Christian Cortez Rojo. Very nice talk, Dr. Chan. Is mitochondrial biogenesis also decreased during development after early exposure to high nutrient diet? Does this counteracts the ability of the brain to counteract mitochondrial dysfunction during fetal exposure to high nutrient diet?
Well, see, we developed several different models. We have high glucose model, high fat model, and high fructose model. I have to say, not all the mitochondrial biogenesis or bioenergetics are being affected by universally. So it's depending on the nutrient or the glucose versus fat or versus glucose or fructose. So it's not simple yes or no answer. I think it all depends on what, as well as the different brain region will have different vulnerability to different enzyme or nutrient exposure. So in terms of in the forebrain hypothalamus, we can see the totally different picture versus the brainstem, the nucleus. So the metabolic perturbation to the same maternal insult could result in different, in terms of mitochondrial function in the different brain region, as well to different impact of different nutrient overnutrition.
Okay, so that answer also relates with the question from Rocio Diaz de la Garza from Mexico. She says, thank you for the talk, and with all this clear-cut evidence, what do you think about the possibilities for regulating the use of high fructose in beverages? There are differences among countries using sucrose or high fructose. Also,
yes, I think definitely the high fructose diet drink beverage. I don't want to name which beverage, but I think it's not good. I try to encourage to young people that don't drink too much soft drinks. Those are really not good for your body. We have tried to but it's very difficult because we try to see this metabolic syndrome. The animal model that the rationale that we develop is use high enough fructose so that we can see the developmental programming of metabolic syndrome in offspring. So based on that, then we do the research and how the 60% high fructose, people will say this is ridiculous, it's too high. Nobody will have this kind of fructose daily intake. But you know, accumulation, if you have lower dose, but accumulate for a long period of time, and we're doing some survey in the in locally here in Taiwan, and see how much sugar or particularly soft drinks with fructose inside the mother take daily, and trying to correlate that with some of the metabolic parameter of their own, as well as their offspring or of their babies in different age. We're in the process of collecting data from this local cohort to see is there any correlation between the intake of sugar, glucose, fructose versus other carbohydrate in terms of their blood level of those biomarkers, if we may call it, as well as in the offspring, when in the young infant as well as a child.
All right, so just to finish the question from Rocio, do you know if there are indications about avoiding these beverages as part of maternal nutrition guidelines in your country or in different countries?
Yes, we do have, but we try to restrict, but I don't know the figure. I cannot give you an exact number, but we do have, particularly for those have a tendency to have high blood pressure and other DM or metabolic syndrome, high lipid level or high cholesterol level. Those mother, we do have a guideline for them to restrict their carbohydrate, particularly fructose soft drinks intake during their pregnancy, and as well as lactation.
Thank you. Okay, thank you very much, Julie. This was a nice discussion. There are other questions also. I will send it to you via email so you can contact the students.
Sure.
Thank you very much. And thank
you
also to all the other speakers, you know, Dan from the Netherlands and Gerardo from Mexico, and all the attendees to this session. Please remember that the next session, session 11, is on cancer, membrane transport and mechanism, regulatory mechanism. We have Dr. Ferreira from Uruguay, Dr. Salomon from Australia, and Dr. Pastor Angra from Barcelona and Spain. So thank you very much, and I will
Thank you very much.
I will see you on May 27, so that's 1 week from now. But the time for that meeting will be at noon GMT. Okay, so 12:00 GMT, not at the usual time, because it's the difference with the Australian people maybe. Thank you very much. Thank you,
Thank you very much.
Thank you.
Thank you.
Bye-bye.
Bye-bye.